biogenic silica, it's magic. Why would I start off a talk about science saying that it's magic? Well, because it partly is. I mean, we know what the substance is that these living organisms are using to build skeletons and protection for their single-celled bodies, but we don't exactly know how they do the patterning. That is still a bit of an active research topic. But I'm going to tell you what I know, and I'm going to let you investigate some of the samples that I brought. So organisms that build with silica. Diatoms, which you can find in that little white powder. Also outside the theater when you walked in, there's a little microscope, and under that microscope are 5 different slides that were created by a diatomist named Klaus Kempt, and he takes diatom shells called frustrules and he arranges them into art forms. I'm getting a little bit of an echo here. Can you hear that? Maybe turn that down a little. Yeah, there we go. And so he arranges those into images, like a little bicycle. I commissioned a Golden Gate Bridge and Coit Tower from him. So be sure to check out that exhibit on your way out if you haven't already. And then adjacent to the exhibit, you can use some laser cut diatom shapes to create your own pictures, and they're very fun to take pictures of with your cell phone cameras. And all of those different shapes and sizes I collected from the UC Santa Cruz online database of diatoms. So diatoms use silica in their cell walls to protect their bodies. Choanoflagellates are colonial microorganisms that also use silica for protection. Radiolarians, which you can see a picture of a radiolarian relative on here. This actually was a drawing that was done by Haeckel, and Haeckel was very enamored with the microscopic patternings of these organisms. And he did lots of really beautiful hand drawings of them, which are all open source right now, which is great. So you can use them in art. And I see a lot of that now. Hexatenellid sponges, that's what I'm passed around, that long basket-like structure that's made out of thin but very flexible silica fibers that are secreted by the sponge. Limpets, those little tiny hat-shaped organisms that are actually snails. They crawl around on rocks in the intertidal zone, and they use their teeth to grind algae, and that's those teeth are made from silica. And then also plants. In the plant kingdom, we have horsetails, which are a very primitive plant, and they are very stiff, hollow-stemmed, and if you touch them, they feel very rough. Sometimes people will use them camping as a scrub pad, a Brillo pad. And then grasses, rice, all the grains that we eat have a lot of silica in their stems. This is an example of many of the patterns that you'll see in diatoms. They're microscopic and very highly geometric shaped. They are specific to the species. So we can tell what species a diatom is by looking at their frustrule. And I also want to say that diatoms are phytoplankton, so they are responsible for giving us maybe half or more of the oxygen that we're breathing. And in certain parts of the ocean that are very, very productive, their reproduction is actually limited by the availability of silica in the water. This is a choanoflagellate. So all of the little circles with black dots inside are the individual cells, and then the colony creates this structure around itself to protect itself out of silica. This is a radiolarian, much smaller than diatoms, and also can be found in some of our local rocks, like chert. San Franciscan chert has radiolarians in it. I once did crush up a sample and try to digest it with acid with the explainers here, and we weren't able to see them because we had a small electron microscope on loan. So we were trying to do that, but I think the preparation required a much stronger type of acid than we had available. And then here's a nice picture of a deep sea sponge in nature. And these can be found in the Gulf of Mexico and also right locally in the Monterey Canyon, underwater canyon. There's quite a few of these sponges. And these sponges are, not only do they create habitat, they create habitat for other organisms as well as protecting their bodies. So in this picture you can see a small shrimp living inside and also a feather duster sea star. And in China, these sponges are very popular wedding gifts because certain species house a pair of shrimps that then live inside of the sponge and reproduce. So online, when I was searching for a sponge skeleton to buy, I found lots and lots of references to Chinese wedding gifts. Here's a little limpet that I took video of in the lab, and you can see its mouth parts. It's on the side of a glass tank, so you can see the little teeth are coming up, and it's sort of almost like a little bit of a wheel. And it's scraping little dots of the algae off of the glass. And then this is horsetails. It looks a little bit like bamboo, but if you touch it, it's a lot softer, and you can kind of crush the stem in between your fingers, unlike bamboo. So for organisms to use silicon in their bodies, it's not in a pure form that Ron was talking about. It's silica, which is what quartz is. Things like quartz will dissolve into water, and when they dissolve into water, they become orthosilicic acid. And so this is the most basic form of this molecule. So you have a silicon atom in the middle, and you have 4 hydroxyl groups all around the outside, so that's 1 hydrogen and oxygen and hydrogen atom, and that allows the silicon to dissolve into water, and then once it's in water, then it can be used and manipulated by organic molecules that are secreted by living organisms. When you have a bunch of silica in the water, you have a high enough concentration of silicic acid, you will start to get just natural condensation and polymerization. That doesn't need to be directed, but it has to be in a high enough concentration. But the organisms have this dilemma where there isn't often enough of that in the water for it to just happen naturally. They have to concentrate it inside their tissues or inside their cells. So there's 2 basic types of biomineralization. One is biologically induced, so just sort of getting the silicic acid into their bodies and secreting polymers or organic compounds, and then it just sort of nucleates and crystals grow. And it's not really directed in any way. And that when that happens, you just sort of get these little nodules of silica. But what I think is even more interesting is the biologically controlled mineralization, where you have these bioseeds and organic compounds, and these proteins and polysaccharides and glycoproteins, lots of different compounds have been investigated that have these properties, and the organisms can arrange them in 3-dimensional space, and then the silicic acid will condense along that scaffold. And that's how these really complex, almost nanoscale structures are made. And then not only do these organisms do this, but they do it at ambient temperature and pressure. So the nanotechnology industry would love to be able to form these very, very elaborate structures at ambient temperature and pressure, and they don't know how to do that yet, and that's why it's still kind of like magic. So these sponges when you're passing it around, you'll notice that it almost looks like a basket has been weaved, and it really is a weave. And at the very base, there's a bunch of very long, straight filaments. Those long, straight filaments are used by the sponge to anchor itself in the muddy substrate. But you can kind of get an idea for how long those strands are that are used in the rest of the body that are almost weaved like a basket to give it a very strong structure. And the patterning of how those fibers are weaved together has been studied by architects, by architects to figure out how to make stronger, lighter weight buildings, because the raw material for that is just a long, thin, hair-like piece of glass, basically. But when you pass that sponge skeleton around, you'll notice that you can kind of touch it and it's not crumbling in your hand. It's got some strength to it. So these long spicules consist of just a monolithic hydrated silica, so that silicic acid that's got the hydroxide groups on there, and it's in a long, thin filament, but then it's surrounded by layers of organic material. So the sponge would normally be growing around that basket-like structure that I passed out. And so you would, sometimes they're almost translucent or almost transparent tissue, and sometimes they're colored like yellowish. And here's a close-up of one of those strands with some of the organic material kind of sheathing it almost and helping guide what direction and where it's growing inside the organism's tissues. So diatoms, they also are using silica to protect their cell bodies, but instead of growing these fibers through their bodies, they are forming these little capsules that fit over their cells. So at the top, you have a single cell. If you go clockwise around the diagram, the two cells separate into two daughter cells first, and then once the two cells have separated their nuclear material, then they need to go about the process of forming two new little caps. And the silica deposition vesicle, or the SDV, is what comes in between those two daughter cells and forms a new cap. And one thing you'll notice is that you're only getting a new cap on the inside. So here's the outside and the inside. They kind of fit together almost like a little pillbox. And so you're going to, over time, get a reduction in size of the diatom cell as asexual reproduction happens over and over again. But there's a different reproductive strategy, that's sexual reproduction, and when the diatoms get too small, then they initiate sexual reproduction and go back up to full size again. Of course, it's all happening asynchronously, but that's, I thought it was a very interesting process of how they are using the size of the daughter cell to then dictate the size of the new cap that they're making. And here's a close-up showing the amazing detail that these have. This is an electron micrograph image. I didn't take this one in the lab. And these frustules are quite varied, and they're used to identify different species, as I had already mentioned. And so because their form carries over from generation to generation, we know that there's a genetic component to this. So we know that the genes are dictating how these organic molecules are being laid down in the scaffold that is then silicized. Diatomaceous earth, which I passed around. Some of you may be familiar with it from using it in filters or for getting rid of fleas. But it's the fossilized remains of the diatom. So all of those frustules fall out of the water and they settle to the bottom of the ocean. And when it's still in the ocean, it's called siliceous ooze, which I think is the best term. I had to put that in here, siliceous ooze. And then once that ooze is uplifted and brought onto land, and it can be mined, and then we call it diatomaceous earth. But they're basically the same thing. And it's mostly amorphous silica, which is, you can see when I show you a sample, there's just lots and lots of just powdery pieces of this, but that means that it's not crystallized the way quartz would be. And it's used in insecticides. Grain storage, it's put in grain to keep insects from eating the grain because it can scratch their cuticles and cause them to dry out. It's also a stabilizer of nitroglycerin, so it's used in the production of dynamite. And also abrasives and filtration, so you can find it in toothpaste. And opal is a semi-crystalline form of silica that is formed from diatomaceous earth, from siliceous ooze that falls down into cracks of rocks. And it can be deposited at low temperature, many different types of rocks. And then if it crystallizes in a certain way, you will get all these different types of colors. And there are many different types of opals, different colors that depend on the type of diatom that was deposited, as well as the type of rock and other impurities that are found in there. But the coolest thing, I think, is not only are they beautiful, but they also, where you find opal around the earth gives you a record of past productivity of oceans. So how much phytoplankton was growing in that area, or when that piece of earth used to be in the ocean, below the ocean, in the geologic past. And this is the structure of precious opal, which will bend wavelengths of light and make all those beautiful fiery colors. But when you are shopping for opal, I learned this because I actually was going to wear my opal earrings, but I forgot today. But I found out that there are certain ways to make the opal look much more impressive by layering a thin layer of the opal on top of a black stone. That will help the light bounce off more, and that those are much less valuable than something that is a thicker opal. So when you're looking for jewelry for gifts or for yourself, you should make sure that it's not laminated. So moving on to the plant kingdom, if you were to take a bunch of wheat straw and burn it so that you had 100 pounds of ash, a big sack of ash, you would have 65 pounds of that would be silica. That is a lot of silica, and that's the hay. If you were just to look at the grain only, it's only 1.5 pounds of silica. And so different plants have different amounts of silica that they will deposit in their tissues for structural purposes, and also to guard against disease. So organisms will land on plants and try to get inside them and grow, bacteria and viruses and fungi. And so silica in the tissues of plants will help protect them, and also against grazing as well. So lots of the grass family, which is eaten by grazers, also is very high in silica. And so this is a piece of slag. If you were to take a bunch of grass and burn it, you could get this silica slag. And I was just talking to an artist friend of mine tonight, and he said, oh, I wonder if you could take that slag and then turn it back into glass. And so you would be able to, take plant material and create stained glass from it. And I said, well, that sounds like an excellent project, but not for me, right yet at least. And these are a bunch of really nice pictures where they've labeled the silica that is in the different tissues of the horsetails. And these cool structures right here that look like little mouths, those are the stomata. So the stomata are where plants open and close these passageways that allow gas exchange. And so during high temperature, they might want to close them off. Low humidity, they want to close it off. But then when it's sunlight out, they want to open it up to get that carbon dioxide in there. But when they open it up, other things can get in there, like fungal spores and viruses. And so it not only helps give rigidity to the cells that are opening and closing, but sometimes they can fortify themselves. And like if there's a damage, a physical damage to the leaf structure, they can then, secrete more silica around that area to protect it. And then, lastly, so the silicic acid is transported from the roots and then up into the plant. So you're getting this material on here, on this slide. Everything on the left-hand side of the line is all talking about silica in the soil and then going from the soil moving into plants. And on the right-hand side are benefits to another organism that uses silica, which are humans. There's been a lot of, this is from a paper that has been sort of a survey of all the different uses of silica to benefit both plants and humans. So some of the things that silica protects against in plants are exposure to high temperature, UV light, heavy metals, diseases like fungi and pathogens. It can help with alleviate metal toxicity. So if you put a lot of silica into the soil, it can help protect the plants against metal toxicity. And then this bottom area is showing you where this silica is coming from that will be utilized by plants. So the solid phase, you have like quartz, clay minerals that Ron talked about in his talk, crystalline forms such as opal, biogenic forms, which are phytoliths, which are structures in plants, and then you would also diatoms or would be part of that if you were to put diatomaceous earth on your garden. And then the liquid phase, once this silicic acid is formed, it's absorbed into the roots and then distributed throughout the plant tissues. So when we eat plants, we get a very good source of silica. When we eat animals or animal products that have plant-based diets, we get a very good source of silica. Or when we drink water, because there is silica dissolved in natural tap water. As well as dust. So if you're breathing in dust or eating dust, if you are using diatomaceous earth in your house, you want to be a little careful about that kind of stuff, because there is a disease called silicosis that can damage your lungs if you're breathing in dust that is high in silica. But diatomaceous earth, I think the particle sizes are a little bit too big to cause that disease. So some of the benefits are to protect against cardiovascular disease and to strengthen bone and other collagen-based tissue. So Ron alluded to that it's utilized in our collagen, which is almost ubiquitous throughout. Tendons, ligaments, all are keeping our skin nice and firm. And so some of the supplements that you can find on the internet are often sold as anti-aging dietary supplements. And I was very intrigued by it to the point where I bought some, and I'll let you know, maybe the next talk that you see me give, I'll look a little younger, I don't know.
Thank you, Denise.